not every day that your sister gets married, but it is every day. Well, actually, like every sub-second that amino acids are getting married in your body in the process of translation to make proteins. So, before I have to go for pictures, let's talk about translation. The recipes for making proteins are written in the language of DNA, and each recipe is a stretch of DNA called a gene. In order to make a protein from that recipe, copies of it are made, written in RNA. So these are messenger RNA copies. And then protein making complexes called ribosomes use those copies as instructions for making the protein in a process called translation. And so that's what I want to talk to you more about today. What happens in translation? So, to understand this, first of all, what's a protein anyway? Proteins are long chains of letters called amino acids. These amino acids have this generic backbone part with this nitrogen hydrogen amino group and this carboxyl group. So a carboxyl is a C double bonded to an O and attached to an OH. And so this generic backbone part allows them to link together. And then they have these unique side chains or R groups, so that stick off, kind of like charms in a charm bracelet. And so amino acids link together through these bonds called peptide bonds. And these bonds are special because they're really strong, but they're also like restrictive because of this thing called electron resonance. So don't worry about that, but just know that there's limited rotation around the backbone part. So that helps give protein some sturdiness. And so you link them up. We call the amino group, and then the free amino group is the N terminus, and the free carboxyl group is the C terminus. And so the amino acids are linked together, N to C. So then you get this long chain of amino acids. And each of those unique side chains or R groups, they have different properties. So there are 20 common genetically encoded amino acids, and some of them are small and flexible. Some of them are big and bulky. Some of them like being around water. Some of them the water hates. So this is going to influence how the protein folds up. So the protein basically folds to try to accommodate all of their different needs, and this gives you this functional 3D protein. But at the heart of all of that is what order those amino acids link in. So each protein has a different order of amino acids and a different number of amino acids and what amino acids are in it and stuff. And so we call that chain of amino acids. So the sequence, we call that the primary structure of the protein. And so how does the ribosome know what order to put the amino acids in to make the protein? And so the instructions are written in mRNA, but that's in the language of nucleic acids. So DNA and RNA are what we call nucleic acids, and we need to get them into the protein language of amino acids. And so to do this, we need to translate. So we're translating from nucleic acids to amino acids. Those different languages. And so this is the basic overview of the process, how you normally see it represented. And so let me just walk you through it. So don't worry about all these words for now. But you have this. So the ribosome is made up of protein and RNA. And it has these three different sites, E, P, and A. And the mRNA, it sits on the mRNA, so the instructions. And then you have these molecules called TRNAs, which are going to bring the amino acids to be added. And they have a matching sequence. They have this thing called, three RNA letters spell one protein letter. And so those three letter groups of our mRNA are called codons. And the tRNAs, so these molecules that are going to bring in the amino acid to be added, they have a complementary sequence called an anticodon. So the anticodon that matches the codon is going to and has the corresponding amino acid comes in. They link together, and so it comes in with the help of this thing called an elongation factor, and the elongation factor spends energy to help make sure that the right tRNA gets there. And then the tRNA is added to the growing chain. Things move over with the help of other elongation factors, and this keeps going on and on until you reach a release factor. So one of the codons spells stop instead. A couple of the codons spell stop instead of an amino acid, and this causes a release factor to bind instead of a tRNA, and the peptide chain gets released. So that's the basic overview in the normal way of looking at things. But I like to think of things a little in a wedding analogy, which is incredibly fitting for today. So I'd like to think of translation as a series of continuous weddings between amino acids. So I'm going to show you the normalized version like I saw you before on the right, and then the bumbleized version on the left. And so we have that ribosome. So that was our complex of proteins and RNA. That is where all of this amino acid joining is going to take place. And so each of these amino acid joinings we can think of as a wedding between amino acids. So this peptide bond formation is this wedding. And the ribosome is our chapel where this is going to take place. And we're going to think of it as a drive-through chapel with 3 parking spots, so the EP and A. And then it's traveling along this messenger RNA road, which has our protein instructions. What about our tRNA? So those are the molecules that have the anticodon that matches the codon and which have the corresponding amino acid. And so that's our link between the mRNA and the protein languages. And so we're going to think of these as limos that are taking amino acid bridegrooms to the ribosomal chapel to get married. So different amino acids travel in different limos. And those limos, we can think of the anticodons as their sort of license plate. And then the codons are reserved parking spots for that specific license plate. And the order of the codon spells out the order in which to add the amino acids. So it's kind of like the order of the reserved parking spaces is going to determine the order of the limos and the protein that's going to be formed. And then we have our elongation factors. And so those are kind of like our escorts, which are going to take the limos to the chapel and make sure everything's all good, and kind of like security, make sure the right ones get there, and then move things along in a bouncer roll. And this is going to keep going until the release factor comes and cuts things off. So in a little more detail, so our ribosome has these 2 main parts, and so it's different in bacteria, so prokaryotic, and eukaryotic, so things like humans have more complicated ribosomes. But there are 2 main subparts. So there's the large subunit, which is called 60S in humans and 50S in bacteria, and the small subunit, which is the 40S in humans, 30S in bacteria. And if you're wondering what those numbers stand for, there's this thing called Spellberg units. So basically, in this ultracentrifuge, you can spin molecules in the sugar gradient, and then the bigger things will sink differently than the smaller thing, and then that can be used to give an idea about size, and the units are the Spellbergs, and so that's where the S comes from. So don't worry too much about that, but just in case you're curious, because it might bug you. And so these are like this large subunit and the small unit are separate, and then they come together. And they have these 3 sites. So you have the E site. So you have the growing chain is held in the P site, which is in the center. And then the tRNA, the entering tRNA come in the A site that gets transferred to the chain on the P site. And the tRNA is going to leave from the E site. And then the mRNA is going to bind between the ribosome's 2 subunits. So how about our tRNAs? So the tRNAs are our limos, remember? So different amino acids have different designated limos, and then they're different with different anticodon license plates, and that match different 3-letter codon parking spots on our road. And so you can, to get like a guide of which one's which, you can look at a thing called a codon table. So basically, each codon specifies a single amino acid, but some amino acids have multiple codons that specify them. For example, if you look at the upper left corner, you'll see that phenylalanine, PHE, has multiple codons that spell it. So we need to make sure that the right limo takes the right amino acid, so that the right tRNA is linked to the right amino acid. So this takes place with the spending of energy in 2 steps. So there's amino acid activation and then tRNA charging. And so in the amino acid activation step, you use ATP, which is a form of cellular energy. to to kind of pay for the amino acids ride into the tRNA. So allow it to get in the right limo. So ATP is, we think of it as a form of energy because it has these three negatively charged phosphate groups that are clamped together, kind of like a clamped spring. And so the negative charges don't like to be near each other, so they repel each other. And so holding them together in that ATP group is energetically expensive. So if you break it, you release energy. So you take that ATP and you break that spring and just put the AMP, so adenosine monophosphate on it. Then you have this pyrophosphate, which can split even further. So you get this release of energy, and you've added a phosphate group to the amino acid. We've added the AMP to the amino acid. And so now you've invested some energy, and so you really want to make sure that things are going to go right. So that amino acid activation is taking place in this molecule that makes sure it helps make sure that it has the right amino acid, because it has these like proofreading pockets that can make sure that it's bound to the right one. And then it also binds to the corresponding tRNA, and so it has specificity for that tRNA as well. So you're having specificity for both the amino acid and for the tRNA. And so this with this combined with the spending of energy helps make sure that you have this you're invested in making sure that you have the right amino acid, because once the tRNA gets to the chapel, it's only going to, the specificity comes from the codon anticodon matching. So basically, if the right anticodon comes, it'll stick to that codon. And so that's all taking place through that the RNA part of the tRNA. And so we want to make sure that the right amino acid is there. And so this is where the amino acid activation and tRNA charging steps, that where the proofreading comes in, really is really important here. And so basically you have that adenylated amino acid, and so the amino acid is attached to the AMP, and it's going to get attached to the corresponding tRNA, and you release the AMP. So now you've paid for your ride, and so you have this tRNA loaded with the mRNA, and so now you want to make sure that it gets to the right place. So the steps of translation. So the first step is kind of different than the rest, and it's different in eukaryotes and prokaryotes. So like humans versus bacteria. So bacteria is easiest to explain. So the initiation step. So initiation always occurs at the codon for methionine. So the codon for methionine moonlights as a start signal. And so basically, if this AG codon comes up in the middle of a protein, then it's just added like a normal methionine. But if it's at the beginning, then it can signal start. And in bacteria, what happens is that you have a special initiator tRNA. It's a modified methionine, and when it kind of like the ribosome assembles around it. So the small first, and then the large comes in and stuff. So you have this initiation step, and so the details are a little nuanced and that sort of thing, different in eukaryotes. But the important thing to know is that the initiation step is different than the elongation step, and so elongation is what comes next, and that's where you just keep adding to this growing chain. And this part is more the same throughout the whole process. So it's just different tRNAs getting added based on the codon anticodon match. So we have that loaded limo, so our amino acid charged tRNA. And so in order to get to the ribosome, so it's going to, so its job, so remember it has that anticodon, which is like its license plate. And the mRNA has these codons, which are like reserved parking spots for that specific license plate. And so in order to make sure that the right limo gets, so in order to get the limos to their parking spots in the ribosome, they take this escort called an elongation factor. And this elongation factor is going to use energy as well. So it uses energy in the form of GTP, which is like ATP, except it has instead of the A, it has a G. So it's just a different RNA letter. And different nucleotide letter. And so when it's so it takes this escort. The escort takes our tRNA to the matching codon. And so basically, if the codon and the anticodon match, they are going to stick together because through base pairing. And so, but if they don't match, they're not going to stick, and so they're just going to like fall back off. But if they stick long enough, then the elongation factor realizes that things are all good, and it's going to split that GTP to make GDP. So it's going to kick off the phosphate unit, and that's just like going back to our spring analogy. We're releasing energy, and so when this happens, it actually causes the elongation factor to change shape, and then it no longer wants to bind there, and so it falls off. And so here we have more energy investment, making sure that the right codon-anticodon pair. So before we were making sure we had the right amino acid tRNA, here we're making sure we have the right tRNA codon match. So once again, we're using energy to pay for specificity. And this is all happening in the ribosomal chapel. So what happens now? So we have our elongation factors come and it's dropped off this amino acid. And now the next thing is we need to transfer it to the growing chain. So we can think of the ribosome as kind of having this chapel, this chimney, where the growing chain is coming out of the P subunit. So our tRNA is coming in the A subunit. And it's going to attach to the growing chain that's connected in this P subunit. And so actually the chain, what happens is the new amino acid is going to attack the chain, and the whole chain is going to get transferred to that new amino acid, and then you're in this really awkward state. So another elongation factor is going to serve as a bouncer. So it's going to come in and get the limos to shift over a spot, clearing the alter. So they're moving together into the P site, and now you have that one that was in the P site is now in your E site where it can leave, and your new codon can enter the A site. So basically now you have a new codon showing up in that A site for a new tRNA to come in. And so this elongation factor uses energy as well. And so there's in the form of GTP with that whole same split and change shape leave thing. Okay, and so you keep doing that over and over in the elongation stage, and then finally you need to end the wedding. Like you've had a bunch of couples, and so it's all this giant polygamous marriage. But now it's chapel closing time, and so the end of a protein is specified by a codon called a stop codon. So there's actually 3 of these: UAA, UGA, or UAG. And so when this stop codon enters the A spot, so when I say enter the A spot, basically remember we keep adding and shifting and adding and shifting and adding and shifting, and so now you've shifted to where there's a stop codon in the A site. And so instead of having a tRNA that binds, it has this protein called a restriction factor, and so it's kind of like a decoy it's a decoy car, so it kind of looks like a tRNA, but it's not. And it specifically it likes the stop codon, and so it's going to come in, and it's actually going to cut off the peptide chain in what's called hydrolytic cleavage. So we call it hydrolytic cleavage because it's using a water molecule to cut. And so when it does this, it releases the growing chain. And so that is how you make a protein. P.S. I'm super happy for you, Nicole, and thank you so much for choosing me to be a bridesmaid. I love you and wish you the best future ever.